Good morning, good evening, good afternoon. Welcome to the second seminar for the International Plant Breeding Series this fall. Thanks so much for your attendance from wherever you're joining. Have the pleasure to introduce Professor Nigel Maxted from the University of Birmingham in the UK. He's going to be talking about genetic diversity and maximizing the founding resources for plant breeding. Professor Maxted, as I said, works at the University of Birmingham. He is a specialist in plant genetic resources conservation and has served several roles both at the European level and at the global level. He got his Master of Philosophy and a PhD from the University of Southampton in the UK. He grew up actually in the UK, Cornwall and Kent, but he has spent some time here in the US and in Canada when young, working in farms. So he knows quite a bit of the US. The other detail about Professor Maxted is that he enjoys, he lives on a farm and he enjoys painting, and the introductory video that I just played has some of his paintings, and he said that he's not very good at it, and I sort of disagree with that. I crossed paths with Professor Maxted earlier this year. We worked together on a project, and I look forward to meeting with him in person one day and exchanging a lot of notes that we had to exchange. So Nigel, I'm gonna stop sharing here, and I'm gonna give you the floor to share. Go ahead.
Right, thank you, Carlos. So as you've heard, my name is Nigel Maxted. Hopefully I'm a much better academic than I am a painter, and I guess you'll be able to assess that at the end of this talk. So the talk is about maximizing genetic resources for plant breeding, and it's in essence, it's why plant genetic resources are important for plant breeders. So what I'm going to cover in the talk, I'm going to talk about the policy context for plant genetic resources, conservation and use, why plant genetic resources are important, how we do plant genetic resource conservation, whether or not we're being effective, and really it would be interesting to hear from you how effective you think us as conservationists are in supplying you as breeders with the material you need. And then I want to finish up. I haven't got time to go through a lot of research, but I've got one research study I wanted to highlight at the end that's just coming to the end of a project, and I wanted to talk explicitly about that. So first, for those of you who are not that familiar with genetic resources, I've given a few references here. A couple of them you can download. The first two from FAO. One is the State of World report, which is a statement of where we are in terms of genetic resources conservation at the moment. And the second is about primarily ex situ. It's if you're conserving material in gene banks, basically how you do it. And if you, this is total self-interest, I assure you. If you want to actually read a textbook on plant genetic resources conservation, then I recently published one for Cambridge University Press. There aren't many out there, and maybe this is not the best, but it's one of the ones that's available. And then I've also given you here a couple of websites which again have information that you can download easily on plant genetic resource conservation and use. And the image on the right hand side here is just to illustrate the fact that if we are going to continue having bigger, better sugar beet, then we need to look for broader genetic diversity, and that diversity may in this case come from wild species growing on a shingle bank in the Mediterranean. So this is the wild relative of sugar beet, which sugar beet was domesticated from. So basically, the problem, why we're having this talk, why we need to have plant breeding, why we need to have plant genetic resources, is to basically feed the human population. And the problem is the human population is rising, not exponentially, but very fast. And so at the moment, I checked this morning, and it's 7.9 billion human beings on the planet. and 78% of those live in developing countries. I'm sure you're all aware of the estimates for how many people will be on the planet by 2050, but it's estimated about 9.8 billion, and 86% of those will be living in developing countries. So a very large number of human beings. All those human beings require food. And so FAO has estimated that in 2050, to feed the human population at that time will require globally a 60% increase in food production. Now, this is not spread evenly across the globe. So countries like the UK and the US, I guess, are going to be, won't have to increase their production that much. But developing countries may, on average, will have to increase their production by 100%. And countries like Nigeria are going to have to increase their production by 160% if they're going to be able to feed the people that are in their countries in 2050. At the same time as requiring more food to feed the people in 2050, the climate change is going to reduce, it's estimated, the IPCC has estimated, they're going to reduce the agricultural production by 2% per decade. So this is really a problem. And you could say that the Earth is beyond its carrying capacity for human beings. So although these little toddlers are very loving things to have around, there are basically just too many of them. So as I said, the Earth is beyond its carrying capacity, but by careful use of genetic resources and good plant breeding, we should be able to keep humankind available and alive for the future. This conundrum of how do we feed this ever-increasing population and the importance of plant genetic resources are recognized in the policy context. So here we have various documents from the CBD and the UN Sustainability Development Goals, each highlighting the importance for genetic resources. And genetic resources feed directly into the UN Sustainable Development Goals 1, 2, and 3, particularly 2.5, which calls for the benefit of crop wild relatives. So these are the wild species of crops. And you get on the right-hand side here as an example of a wild relative of garden pea, which is growing in the Caucasian mountains in the Caucasus, over 2,000 meters. This is the wild Vavilovia formosa. The UN also, in the CBD, set up these 5 key indicators, and one were the trends in genetic diversity of domesticated animals, cultivated plants, fish, and other species of socioeconomic importance and their relative conservation. So if we look at the 5 key indicators, 4 were not met by 2020. The only one that was still not met, but better met than the others, was the one in conserving genetic diversity, which is directly linked to food. It's the one, it's not good, but it's better than the others, which were complete failures. So if we look at agrobiodiversity, what is it? So this is the element of global biodiversity, which is associated with agriculture. So if you look at these spheres, then we have global biodiversity, a lot of species globally estimated that in terms of described taxa, about 15 million described taxa thus far, and maybe another 100 million yet to be described. A proportion of those are impacted by biodiversity, by agricultural cultivation, and then a smaller percentage of those are associated with agrobiodiversity, and they provide ecosystem services for humankind. And then a subset of those, the inner sphere, are about 7,000 domesticated species and 60,000 wild relatives of those 7,000 domesticated plant species that are actually cultivated. So in terms of crops, as I said, the total number of plant species are about 340,000, of which 30,000 are edible. And as I said, 7,000 are cultivated, about 3,000 are cultivated for food. And then the wild relatives associated with those. But we all know that it's not 7,000 that are cultivated widely. It's a much lower number. So 30 crops cover about 95% of calorie production, 12 crops, 75%, 4 crops, produce 60% of calories. So one of the things we're really worried about is there is such a focus on such a small number of crops, and we're continually trying to expand, as pointed out by Darwin himself in 1868, that we need to have a wide array of crops, a wide array of varieties, and each variety needs to be genetically diverse as well if we're going to be able to sustain production. And just for comparison, I put in here the figure for animals. So here we are talking about 70,000 cultivated crops. In terms of animals, we're only really producing 50 non-carnivorous mammals and 10 bird species. And the wild relatives of animals are not really have any substantial genetic diversity, which is used in breeding. So there's a big difference between animal and plant genetic diversity and the agrobiodiversity that's used in breeding programs. So what are genetic resources? Genetic material of animals and plants, which are of value as a resource for present and future generations of people. And to illustrate this, I've given obviously a shot of a contemporary wheat. You know, wheat is a hexaploid made up of triticum turgidum, and interestingly, the other genome, the other third genome, the D genome, comes from torchii. And torchii is a small grass that you would never think is that really important. But it really is, this grass that you find growing wild in the Mediterranean is something that you would never think has the importance it has if you see it in the wild. But the point I'm making is that often with genetic resources, they're not obvious that there's something that would be of value to maintaining humankind. So the importance of genetic diversity, as I was saying, we need to increase diversity at those three levels in terms of the number of crops we cultivate, in terms of the number of varieties, and also the diversity within varieties. If we reduce that diversity, then we run the risk of disasters in terms of loss of production. And there are many, many examples in the literature. I won't go through them. But really, crop and varietal diversity is key to the future of farming and the subsistence and global food security. So what are the aims of me as an agrobiodiversity specialist in terms of conservation? Well, our range is just as I said, to maximize the broad range of genetic diversity in the 7,000 species we're trying to conserve and make available to you as breeders to be able to use. So what are we actually aiming for? This has been characterized at the genetic level as 95%, which is most, of all alleles at a random locus occurring in a target population with a frequency greater than 0.05. So we're trying to conserve everything except the things that are very, very rare, because the likelihood of being able to find those very rare materials is low. So we tend to focus on the other species which are not so rare and try and conserve 95% of those. So this enables us to maximize the potential adaptive diversity that we can then pass on to breeders. In terms of the actual strategies that we use, as defined by the CBD, we have two strategies, ex situ and in situ. So with ex situ, really what we're doing is we're locating the population that we want to conserve. We're taking a sample and then we're transferring it to a distant location and storing it and conserving it at that distant location. So it's ex situ, it's moving away from the original location where the population was found. Whereas in situ conservation is the reverse. You're actually locating your population, but you're designating the area around your population for conservation. And then you're managing and monitoring the population within the protected area that's established, if that's the way you're doing it, to maintain the genetic diversity. You want to see maintenance of genetic diversity, not falling off in terms of population numbers or genetic diversity. For those of you who are really interested in obscure detail, there is a third mechanism, which is called circa situ, which is really, it's a kind of in situ, but it's transferring them short distances. So if you have a population which is in a place where it's really unsustainable, or you need to have ease of access to that population, then you may transfer it a very short distance, but then it's actually maintained as an in situ population. So essentially we have two techniques, in situ and ex situ. And within those two strategies, we have numerous techniques. So here we have the four techniques for in situ. Primarily the ones that are used for genetic resources are protected area and on farm. So let's go through those. So protected area is where you set up a protected area. You have a particular area designated within a defined area to conserve natural wild populations, and it's actively managed. In other words, you're intervening at the site to give priority to the maintenance of your target populations. And the goal is to maintain it for long term. You can't do it for a short term. It has to be long-term conservation. These days, it's quite expensive to do in situ protected area conservation. You need a lot of staff and a lot of time involved in managing the populations. So there is this other, it trips off the tongue beautifully, other effective area-based conservation measures. We must think of a new name for it. But this is conservation outside of protected areas, where the populations can be more informally managed. So an example of this might be if you have management of the species growing under orchard trees or along the edge of a roadside, where the population or the roadside is not sprayed with herbicide or burned or whatever you do to it before it's had chance to set seed. So it's a means of doing conservation on the cheap, if you like, but in that sense, it's much more cost efficiency. And then if you're dealing with crops themselves, then the main form of conservation is on-farm conservation. So here, instead of having a conservationist doing the conservation, you have the actual farmer, and they maintain the traditional animal breeds, crop varieties by the farmer within traditional agricultural, horticultural, or agro-silvicultural systems. A slight variant on that is where you have people in their back garden who grow a lot of species for home consumption. So that's home garden conservation, which is kind of an on-farm conservation, but generally has more diversity in terms of crops grown, but very small population sizes because the material is grown for home consumption. So in terms of ex situ, again, we have several techniques that are available. But the primary one, which has over 90% of conservation, I guess, is seed storage. And this is where you collect your sample of seed. It's an orthodox sample, which means if you store it in a gene bank, it doesn't actually kill the seed itself. Some species, generally species that are very high in fats, if you try and dry them and store them at -18, then it actually kills the seed and the plant is dead. So that's what we call a recalcitrant species. So orthodox species, which is most species, can be stored in a deep freeze. So basically you dry them to about 15% relative humidity, and then you can store them at -18. Increasingly these days, it seems to be cheaper and more efficiently to cryogenically store them at -196. And then if you have recalcitrant species, then you would use usually, these are the ones you can't store in a gene bank because it kills them, then you would use in vitro or filled gene banks. There are advantages and disadvantages for each in situ and ex situ. So here are the advantages for both. So in situ is obviously suitable for all organisms because you're conserving them where the population is already found. It's dynamic, you have continued evolution. They're able to evolve alongside the species and pathogens. You can also do evolutionary or genetic studies in the field, suitable, as I said, for recalcitrant species. And if you have your target species trying to conserve might be, say, a wild barley, but in the same location, you may have other species. So you have this possibility of additionality in the conservation. Whereas if you're looking at ex situ, it's highly reproducible. Ex situ using a gene bank can be used for, as I said, probably 90+% of species. They can be stored in there for a long time, hundreds of years, potentially even thousands of years, though no one's actually tested that yet. You can conserve a lot of diversity in a relatively small area. If you want to do evaluation and breeding, it's fairly easy to take samples out, defrost them, and put them through evaluation or breeding. And the actual cost of maintaining a population within a gene bank is only estimated to be about $5 per year. So it's relatively cheap. The point is that don't put all your eggs in one basket, in that you don't just use one technique for conserving your target species. You tend to have possibly one ex situ and one in situ conservation technique. And to illustrate why this is important, this is the photograph is a particular peninsula just off the coast of Turkey, Western Turkey. And on this peninsula is a small wild relative of wheat, which is quite rare, relatively rare. It's an Aegilops species, which is just found on this peninsula and other locations, but this is one of the best populations. Now you can see how close this is to the water. With climate change and rising sea levels, that population is highly threatened in the near future. So we want to have complementary conservation. It should be conserved, material should be in the gene bank, and in this case, probably focus your in situ efforts in another place other than this one, even though it's the best current population. So what we're looking for when we're doing our conservation is adaptive diversity, and adaptive diversity is diversity that can be used. It has advantageous traits that can be used by you, the breeder. And so this just illustrates the different parts of agrobiodiversity that have the maximum amount of genetic diversity. So you start off with the wild species. You can see here they've got a lot of different colors. It's domesticated, and immediately you're losing a significant amount of genetic diversity just through the process of domestication. And then land races are still, within crop terms, the most diverse element of the crop. But if you narrow that down to modern varieties, then again, you're narrowing the genetic diversity. And most modern varieties are homozygous, genetically uniform. So in terms of conservation, again, the efficient place to do most of your conservation is in wild species, because that way you gain your maximum diversity for your conservation effort. And Tanksley and McCouch in 1997 published a paper which showed this diagram and the argument for conserving wild species. And they talked about, for tomatoes, for instance, 95% of the genetic diversity for tomatoes is in the wild species of Lycopersicum and Solanum. So let's look at what is actually being conserved. This is some data taken from the second State of the World report that was produced in 2010, produced by FAO, a statement of what's happening in terms of conservation. And they estimated then that we're about 7.4 million accessions of agrobiodiversity seeds, plant diversity seeds in gene banks, and about 1.4 million had been added since the first State of the World report that was produced in 1998. So in that 12-year period, we had an additional 1.4 million accessions added. It's estimated by Subster that 70% of genetic diversity of the top 200 or 300 crops is already conserved in gene banks. Personally, I doubt this figure, but it is a figure that is put forward by Subster, and maybe I'll explain why later on. This map at the side shows the larger gene banks around the world, and you can clearly see here the largest gene bank at the moment is Svalbard. So Svalbard, you've got a shot of the front door of the gene bank on Svalbard Island, which is on the Arctic Circle. It's a Norwegian island, which is setting up this global repository of safety backup for humankind of genetic resources. So that's basically looking at ex situ conservation of plant genetic resources. Then we have this almost like a footnote at the bottom here, which says very limited long-term in situ or on-farm conservation. And again, I'll come back to talking about that later, but there are only about 5 or 6 places around the world where you have active. By active, I mean you're actually monitoring and managing the site, in situ conservation of crop wild relatives within protected areas. And in terms of on-farm conservation, although you have many farmers obviously maintaining land races, to have a secure maintenance which is maintained for the long term, generation after generation, there are virtually no examples of that being guaranteed so far. So this is something we need to build on for the future. So definitions of the main elements of agrobiodiversity, plant genetic resources, which we're dealing with if we're dealing with conservation, are land races and crop wild relatives. So first of all, land races. These are traditional forms. They have numerous names, as you can see here. And Harlan in 1975 described them as populations that have evolved from subsistence agricultural societies as a result of millennia-long artificial human selection mediated by human migration, seed exchange, and natural selection. So what he's saying is these are natural or man-derived populations where you have annual seed saving if they're annual species, and over that process they're maintained for a very long time by certain communities. And there was that was his definition. Then we did one in our research group, which came from this in the shot. The person on the left-hand extreme is Maria Camacho Villa, a Mexican woman who worked with us on defining crop wild relative land races, etc. And here is her definition. It's a dynamic population of cultivated plants that has historic origin, distinct identity, lacks formal crop improvements, have not been bred by plant breeders, is genetically diverse, locally adapted, found often in traditional farming systems, and often has some cultural association. You find that rice, for instance, in Asia is often given as a present to a couple when they marry to start their life together. Whereas crop wild relatives, these are the wild species related to crops. They form the ancestors of the crops we have today. And they still have, as I showed with that image earlier from Tansley and McCooch, a lot of genetic diversity that can be used in breeding today. So a broad definition of a crop wild relative is any taxa found in the same genus as a crop. So to illustrate that, if you're looking, say, at barley, hordeum, then the crop itself is Hordeum vulgare. Any other Hordeum species is a crop wild relative of Hordeum vulgare. We have a much more precise definition, which uses gene pool and taxon group concepts, which decreases the number of these species and that enables you to focus on the ones that you think are going to be most useful to you in terms of plant breeding in the future. So this is just showing you various shots of land races, including the one in the center is from the Scottish Islands. It's a barley beer. It's actually called beer barley, B-E-R-E, not E-E-R. And this barley, when we started conserving this land race, a lot of the local distilleries were very interested in using traditional barley to make their traditional whiskey. And so now we have a new use or an increased use of that, promoting the land race and adding value to it in terms of whiskey production. It's even used for making beer itself, for proper beer, and a drink, which you can drink the beer tea. In terms of crop wild relatives are found in all countries, as you'll find out later, a lot in the US. So this was an analysis of crop wild relatives in England, you can see in this particular case. And the areas that had the highest level is this one here on the south coast. And the second highest site was this one here. And this one here has recently been designated as the first genetic reserve, in situ genetic reserve for crop wild relatives in the UK. And what is actually the first site that has the most diversity will be set up as the second site soon. And these are two of my students handily pointing out specific crop wild relatives of cabbage and of beet. So what are the threats? Why are we worried? Why am I going on about conservation? Because there are lots of threats that are threatening plant genetic resources at the moment. So land races are being replaced by modern varieties. Agricultural intensification is not allowing weeds to be present in crops, and crop wild relatives are often weedy, found around crops themselves. Changes in land use, human population migration means a lot of these, what would have been weedy fields, are no longer cultivated. Human poverty, starvation again means that you have a lack of cultivation of crops. Land degradation, it becomes unsuitable for growing crops. And then climate change is obviously a very big factor in terms of a threat to agrobiodiversity. So specifically looking at land races, so here's this image on the right-hand side is data from the US. This is looking at the number of land races that were present in 1903. So if we look at beet, in 1903, the number of land races that were present in the US were 288. And if you look in 1983, it was down to 17. If you look at cabbages, 544, and in 1998, 28. So you can see the pattern. All of these land races are shrinking. The number of land races that are available are shrinking dramatically over time. And I perhaps foolishly published a paper in 2008 stating that land races were the most critically threatened element of biodiversity in total. And why? So how do I justify that very radical statement? So basically, we have no idea how many land races exist. The people who maintain land races tend to be older people. In the survey we did on the Scottish Islands, where that beer barley came from, the average age was 65 in 2003. I went back last year to the Scottish Islands, and the number of people now growing land races has decreased by 45%. So it means that those 65 people became older and died, and the material they were conserving has been lost subsequently. So 45% of genetic diversity in land races there was lost over that fairly short 15-year time period. So we don't know why farmers basically grow land races, and they grow them not for conservation itself. They're not conservationists. They grow them for economic yields. So many of them are switching from growing land races to growing modern high-yielding varieties. On the Scottish Islands, interestingly, they can't switch because there's a very unique soil there that won't, the modern high-yielding varieties won't grow in that condition. Many seed companies promote modern cultivars, obviously, to replace land races because they're less productive. And no agency in most countries has active responsibility for conservation of land races, and no country has a comprehensive inventory of land races. So that's how I justify the fact of saying that land races are the most threatened element of biodiversity. And if we don't do something actively about their conservation, then they will go extinct within my lifetime, not yours, and I'm sure you're much younger than I am. Within my lifetime, those land races will go extinct. In terms of crop wild relatives, we did a red list assessment. Probably the largest one done so far is the one done of European crop wild relatives, where we looked at 572 of them, and 16% of those were threatened or near threatened, 4% critically endangered, due to go extinct within the next 10 to 20 years. If we actually look at the amount of material that's conserved ex situ of crop wild relatives in Europe, then only 10% of total conserved collections in gene banks are crop wild relatives, and that those 10% represent only 6% of all crop wild relative species in Europe. So 94% of crop wild relatives in Europe have no material in gene banks, very high percentage. If we look globally, the figure is about the same. 72% globally are under conserved, and only 5% are well conserved in gene banks. So 95% globally of crop wild relatives are poorly conserved or not conserved at all. If we look at in situ conservation for crop wild relatives, you can say that they're present in many protected areas. They are, but there we have some what we call passive conservation. So they're found in a protected area, but it's not being actively managed or monitored to conserve that particular species. So the places around the world, as I said before, there are only maybe 5 to 10 sites around the world where we have active conservation of crop wild relatives within existing protected areas. This is just a nice graphic produced by Colin Huri, which shows that the no single country has a monopoly of genetic resources, crop genetic resources. So different areas of the world is where we find the diversity for different crop wild relatives and their associated gene pools, and where those species were actually domesticated. Perhaps I should just point out that the area of the world with the highest concentration is West Asia. West Asia has more crops and crop wild relatives than anywhere else in the world. Okay, so this is the final slide before I go into the case study. And often when I give this talk, people, and I think maybe it was the sort of question you might ask as well, is why do we need crop wild relative landrace breeding? We already have enough material. Why do we need more? But with genetic engineering, we can produce whatever we want. So why do we need? We do need it, yes, because there are a massive amount of adaptive traits that it's impossible to know what's available. So we need to actively conserve them. So we can't stop going out and conserving material for the next generation because we don't know what will be required, and if we don't do something about it, it will be permanently lost. It's not possible to practically have genetic engineering for all crops. We're going to do it for all 7,000 crops? I don't think so. And also, it's not possible to do genetic engineering in every country of the world. And that genetic engineering is focused perhaps on major genes, does not work for genes which are more complex and require gene interactions. So the answer is yes, genetic engineering is a very good technique, but it's not one that can be totally replace the need for genetic resources. Okay, so I want to finish up by going through a bit of research. That's basically an introduction to plant genetic resources conservation, and then hopefully linking it through to use. So this is a project, Global Crop Wild Relative Project, that was led by the Global Crop Diversity Trust with funding from the Norwegian government. And the focus was primarily on ex situ collecting in relation to breeding with crop wild relatives. So most of the budget was spent on breeding with crop wild relatives, but there was an element to do with conservation as well. So what we did in the project, we looked at the major crops and found they were in 92 genera. We then collected a lot of background data on those 92 genera. In other words, which crop wild relatives were present within them, what was the data, the passport data associated with those, and we did a simple gap analysis. So a gap analysis is simply comparing the distribution of the species which are currently actively conserved, either ex situ or in situ, against the wild natural distribution of the species found in nature if they're crop wild relatives. And from that, we can then identify the goals for in situ and ex situ conservation. So then we did field collecting and ex situ storage, and a lot of the material, because it's done by the trust, and the trust are the people in charge or partially in charge of the global vault in Svalbard. A lot of the material is going on to Svalbard from this project. So we did our looking at the crop wild relatives that were present, and we called the inventory of 1,667 priority crop wild relative taxa from 194 crops, the Harlan and DeWitt inventory, because we needed to use the gene pool concept put forward by Harlan and DeWitt to define the crop wild relatives. So those crop wild relatives come from 37 families. We had 1,392 species, etc. And the data is available from the website listed there. The family which has the highest number of crop wild relatives are legumes, followed by Rosaceae, Poaceae, etc. Perhaps no surprises there. The distribution, as I said earlier, the concentration of crop wild relatives is focused in Northwest Asia. and then following that, China. And then interestingly, you can see the only country on this list that's represented individually, I think, is the US. And the US itself has 152 global priority crop wild relatives present. So that's not the total number of crop wild relatives in the US, but it's the ones that are globally important. So then we can look at species richness with all that distributional data. So we have the 1,394 species. We collected passport data locations where those species have been found, and then we overlaid all those and came up with this hotspot map, which shows species richness. And this is the area, the hotter areas are the areas which have species concentrations. And you can see primarily around the Mediterranean, the richest areas, but interestingly, large populations in the US, which hadn't been recognized before, and also in Northern Australia, which I'll come back to in a minute. We can then compare that richness map against areas of the world where we have material that's already present in gene. Richness here is seen as the areas that are ripe for further collecting. So you can see the areas that require further collecting are in Indonesia, in central China, and Turkey. And interestingly, Portugal comes out of the country with a whole lot more collecting required, which we hadn't previously recognized. So then, as I said, this was from the Trust project, and we have actually, as part of that project, done a lot of collecting. So in 2013 to 2019, we did collecting, and we collected 3,002 seed accessions from 242 priority crop wild relative taxa. The paper has only just come out, Muller et al. And that the collecting was actually undertaken by the Royal Botanic Garden, Kew, working with the CG centres and national programs worldwide. So significant additional diversity of crop wild relative material now in gene banks, but I hasten to stress this is still not going to make a dramatic difference in terms of conservation. There were still large gaps in terms of ex situ conservation of crop wild relatives. The situation, as I said earlier, is much worse for in situ conservation. So here is another analysis where we tried to pick out the best 170 sites around the globe. If we wanted to conserve crop wild relative diversity globally, which are the top 170 sites, both inside and outside of protected areas? And for each species, so that's each of the 1,394 or whatever species it was, we have 5 populations included in this analysis. The 5 populations are not chosen randomly. They're deliberately selected in places to maximize genetic diversity. And the sites that have been chosen have all been tested for climate change to make sure that they'll still be there for the next 50 years. Populations will be able to exist in those locations for the next 50 years. And again, you can see the numbered sites are the higher ones, the earlier selection of sites. And so, for instance, if you're looking at sites within protected areas, then the first site is actually in Spain, and the second site is in Greece, etc. And the third site is in the US. So that's the in situ priorities. And we've called this network of sites the Vavilov, after Vavilov, who was the first Russian geneticist, scientist, who was the first one to recognize the importance of crop wild relative conservation and start actually going around the world collecting them. I mentioned Vavilov. The other thing that Vavilov did is he looked at the distribution of centers so crop wild relatives not found evenly across, as you saw from the globe, as you saw from that map. They tend to be found in 8 centers that were defined by Vavilov in 1926. 1926, this is before we had even the idea of hotspots for biodiversity. So really hotspots were pre-invented by Vavilov specifically for crop wild relatives and land races. And so unfortunately, obviously, Vavilov doing his work in 1926 did not have all the data we have today. So we've recently revised the Vavilov centers and added additional centers. So we added an additional center in Brazil, and we noted the populations, the centers in the US. So both in California and in the Eastern Seaboard. And then a site in West Africa along the coast, and then a site in Tanzania, and then northern Australia, as I mentioned earlier, areas that we did not know previously until we'd done this analysis were hotspots of crop wild relative diversity. So then the last thing, we are getting on to the last bit. So this is the comparison of Myers or IUCN Conservation International listed hotspots, global biodiversity hotspots, which are in blue on this map, and the orange areas are the crop wild relative hotspots. And interestingly, if you look at this, both for the Americas, for Africa and Europe, and for Asia, there is not a perfect correlation between crop wild relative and biodiversity hotspots. There's some sites overlap, but there is a significant difference between those. So if we just focus all of our conservation effort on biodiversity hotspots, we're not going to get good conservation of crop wild relative diversity. Okay, so last slide, just finishing up. I just wanted to make clear that for breeders, there is significant value in terms of food security in using maximum plant genetic resource diversity in your breeding. The estimate for the value of crop wild relative use in 29 major crops was estimated in 2013, and annually it's thought to be worth $115 billion. So that's for the use of crop wild relatives in just 29 major crops, 115 million. So imagine if you used crop wild relatives in the breeding of the top 300 crops, how the price, the value of the additional value as a result of that could be extraordinary. I mean, this figure of 2.3 trillion is obviously a guess, a silly guess. Probably not that high, but still it's significantly higher than the 115 billion that currently comes from their use in just 29 major crops. In terms of the conservation, the GP Global Strategy for Plant Conservation Target 9 was that we would have 70% of plant genetic resources conserved in gene banks and in situ by 2020. We failed, basically. We estimate we've only have about 5% conserved well in gene banks. And as I was saying earlier, virtually no in situ conservation, maybe between 0 and 2% of crop wild relatives and land races conserved in situ. So at the moment, I would argue that plant genetic diversity is limiting your breeding ability. And so we must have much more improved, better supply and better provision. It's not just conserving the genetic diversity, it's making it more easily available for breeders to use. So thank you very much. I'll finish there and we'll be available to answer your questions. So I'll stop sharing my screen.
All right, thank you so much, Nigel. If you want to open up your camera so people can see you. Thank you so much. Very
okay. I hope that was
yeah, thought-provoking presentation. We already have one question in the Q&A from Flavio, and it says that he noticed that the Centers for Wild Relatives, Centers of Diversity, appears to coincide with areas where the most population of the globe are, and what are the implications for that?
So maybe it's not surprising that these species were chosen to domesticate by early man, because they tend to be weedy species that were found around human habitation, as they were then, and they still are today. So the figure I gave, remember I did that assessment of crop wild relatives and the percentage that were threatened, it was 16%. So 16% of crop wild relatives are threatened using the IUCN Red List criteria. If you look at the global figure for all plant species of the percentage threatened, it's actually 22%. So crop wild relatives are more amenable to being around human cultivation and around human habitation. So they're less prone to extinction as a result of human action than true wild species or other wild species, let's put it that way. So it is, so obviously if we totally trash the planet, then the crop wild relatives will be destroyed as well. But the other species will go before the crop wild relatives.
Again, you guys can put the questions in the chat or in the Q&A. I have one observation or request for you, Nigel. It seems to me that a lot of the time we suffer the consequences of our unsuccess when we develop these projects to improve, genetically improve materials and bring them back to farmers, which if they are successful, if the project is successful, well, it ends up making disappear a lot of the landrace diversity because farmers will certainly adopt. We are now
breeding paradox.
Yeah, the breeding paradox, exactly. One of the initiatives that we're working here in NC State with some of our Kenyan colleagues is about African indigenous vegetables. And I've been pushing to include as part of that initiative a module on studying the current available diversity and making an effort to understand it and collect it and preserve it, because if we are successful with this project, we probably will be making disappear quite a few land races. Is that something that is usually done in proposals, project proposals? Is something that you would favor? And I just want to open an invitation for you to help us with this.
So, so certainly it's something I would encourage, and it's something I think, but so it does partially depend on the species itself. So if you're dealing with cucurbits in general are, if I may use the term, barely domesticated. In other words, there's not so much difference between domesticated cucurbits and the wild species. And sometimes you still get gene flow between the two. And so in that case, yes, you should definitely be doing it. I think you should be doing it on all, because you're then creating a resource for use in breeding, but there is less chance of having that kind of natural integration, I think, between other species. Like with wheat, for instance, it would be very unlikely you would get integration these days between wild aegilops species and wheat itself. The other example that is shown is there's actually in the literature is with maize in Mexico, which I've not seen myself, and it does seem to me logical if you have Tienda City growing wild in around maize or corn, why there is this, local people say you get the hybrids forming, but then I think the farmer would actually get rid of the hybrids, because you can imagine if you cross cultivated maize with a weed, basically, which is a degree of magnitude smaller, the likelihood of getting usable cobs off that hybrid plant are going to be very little in the first generation.
Right.
So I don't think the farmer would be willing to take that on.
Sure,
Whereas I said, if you're dealing with cucurbits, there is less difference.
Okay.
But you do often, there is quite a literature about this wild hybrids of Mexican maize with wild maize with cultivated maize.
Okay, we have a question from Mary. I guess I think she's attending from Benin in Africa. And how long would it take to effectively use wild relatives in breeding, and what innovative methods can be applied to breed for those and ensure success?
Right. So how long will it take? So if you're starting with the initial crosses, I guess it's going to take you something like 10 generations to get something reasonable out of it. So generally what happens is these days, the CG centers and certainly the Crop Trust are doing pre-breeding for other breeders. So rather than actually do the crosses yourself, I would suggest you link up with one of the CG centers and use their pre-bred material. So they have material where they've already crossed it with the wild relative, they've got the traits and they've got it past the first few generations. If you then use that material in your breeding programs, you're going to have the benefit of the wild relatives, and you don't have to go through the agony of getting rid of all the other deleterious traits that come across when you're hybridizing with the actual crop wide relative material.
Okay, yeah. Again, you guys can post questions in the chat or in the Q&A. I have a note here that I made when you were presenting. There seems to be some crops that are more vulnerable than others. I recall I made a consultancy in oil palm, and I recall reading that the genetic base of most of the cultivated oil palm comes from 5 genotypes that the Dutch brought to Singapore, and that was the origin of most of the improved oil palm in the world. So I cannot imagine a crop that is in more dire situation than that.
Yes. Well, I'm very pleased to say that it wasn't the Dutch, it was Royal Botanic Gardens Kew who took the original.
Oh, good.
As an Englishman, I guess I have to stand up for my own side. But also to say that even more pleasing is, I used to run in the University of Birmingham. We had, I think it was still at that time, the only MSc course in plant genetic resources. And someone we trained on that course was from Malaysia. And we realized, he came on the course, realized the benefit of having greater diversity, and actually did a collecting expedition back to West Africa to collect the original or more populations and take them to input into the breeding program in Malaysia and Indonesia and other countries in the region. The other thing they did that was absolutely, I wish I could remember the figures in my head, but the other thing they did is oil palm is pollinated by some kind of insect. I can't remember which kind, but the insect is not present in Southeast Asia. So they were producing oil palm, you know, the actual, like the dates of the oil palm, without having the pollinator. Once they realized this and took a pollinator to Indonesia and Malaysia, they increased production by something like 70%. Maybe that's an exaggeration, but it was an absolutely phenomenal amount of additional productivity from taking the pollinator and getting that much more seed set on the, or inflorescence set on the head of the oil palm. So that was really very, very dramatic, and again shows how important genetic resources are, I think. Yes,
definitely. Mary, I see your hand is up. I don't think I can open up the mic for you to ask a question, but if you can write it in the chat, that will be great. We talked the other day, Nigel, about the need for more formal courses, particularly for graduate students in genetic conservation and use. I see that as a tremendous need here in our system. There's a few universities that offers not degrees, but just courses, okay?
Yeah.
Are you better off in the UK and in Europe in general?
Well, I would say this is a particular thing for me because, as I said, I ran the only MSc course I think that's actually existed entirely in plant genetic resources. And the course ran from 1961 to 5 years ago. And the course closed simply because we had a lack of studentships. So we'd had studentships throughout the whole of that time from FAO and the forerunners of biodiversity. It stopped, which I think was not a mistake. I don't understand why they did it. Is, you know, why bring students from Africa and Asia to Birmingham to do an MSc if they're kind of ridiculous? It should be taught in Africa and Asia. I totally agree, but there has to be that training. And so we had, if you look around the world at the people in genetic resources, I would say probably, well, certainly more than half of them, the older people went through the Birmingham course because it was the only course. And I think now there is a general lack among younger people in the field of expertise in plant genetic resource science. So I definitely think there is a need for it. And I think if you're teaching breeding programs, I strongly believe you can't teach breeding if you don't understand what's actually happening. And that's why, in a sense, the talk I gave was slightly patronizing. It was a fairly low level talk. I wish I could have talked more about the research, but because you can't assume these days that most graduate students have a good knowledge of plant genetic resources, as well as breeding. And there are so many breeding techniques as well, many breeding techniques to learn. It takes time. So, I think there is a real need for genetic resources teaching. I completely agree.
Okay. Well, thank you so much, Nigel. I'm very grateful for your time and for your seminar, okay? I wanted to thanks the attendance, and the seminar recordings will be, the link will be shared pretty soon. There's a lot of people that expressed to me that they will not be able to attend today, but they will certainly look at the, listen to the recordings. Credits to the music that was played at the introduction is there, and special thanks to Brandon and Anthony for helping with all the logistics. Next Thursday, we're going to have Dr. Stephen Smith, former Corteva DuPont pioneer breeder, talking about estimating the global usage of corn then germplasm and the implications for the sustainable use of made genetic resources. So Nigel, you're specially invited to this seminar because it's right on your field again, okay? So thank you so much, everybody. I hope you guys have a good rest of the week. Thanks for the attendance. Thank you, Nigel. For
thank
your
you.
time.
Thank you.
Okay.
I hope you enjoyed it.
Yes, very useful. Thank you so much.
Okay, bye.
Bye-bye. Bye.